Hello, everyone, and thank you so much for the opportunity to present some of the recent work in our lab. To give you a brief background, we have previously shown that endothelin-A receptor antagonists significantly increase opioid analgesia, and in subsequent studies, we also found that analgesic tolerance as well as withdrawal symptoms are attenuated by these antagonists. This was one of the most important findings from our previous animal work, but we wanted to go a step further and investigate the mechanistic role of some of the signaling pathways in cellular models in how BQ123 mediates its effects on morphine. So in the present study, we used neuroblastoma SHSY5Y cells, which express both the mu opioid receptor as well as the endothelin-ETA receptor. We created a stably transfected cell line using the cAMP-GloSensor plasmid for cyclic AMP, the cyclic AMP23F plasmid specifically. And after a series of transfection optimization and after the stable pool of cells were created and maintained, we further then optimized the GloSensor real-time cyclic AMP detection assay in these cells. In acute studies, our treatments with morphine, BQ123, and a combination of the two were done for about 2 hours. We also used isoproterenol, an alpha adrenoceptor agonist, as a positive control. We were excited to see that under acute conditions, morphine challenge doses produced a significant decrease or an inhibition in the cyclic AMP response as measured by the relative luminescence units as shown in figure 2. This is the acute treatment with morphine. We then looked at what was the response to acute BQ123 treatment without the presence of morphine. And we were very intrigued to see in figure 3 that acute BQ123 treatment, when we looked at a series of concentrations, a range of concentrations, we saw that there was actually an increase in cyclic AMP response. This is something that we are further investigating, and we will be excited to share those results with you in the future. In chronic studies, morphine as well as BQ123 treatment was done for a 24-hour period. And interestingly, in figure 5, we saw that with chronic morphine treatment, without the presence of BQ123, there was an increase in cyclic AMP responses at higher concentrations of chronic morphine. This is indicative of tolerance in the cellular model, similar to how we see longer latency responses in animal models. The other interesting finding was actually, to draw your attention back to figure 4, was when we looked at the acute combination treatment of morphine as well as BQ123. It was interesting to see that the cyclic AMP responses were further inhibited in the presence of BQ123 when a challenge dose of morphine was given. And this again produced a concentration-dependent decrease in cyclic AMP in response to morphine. So this was another very exciting finding, which helps link some of the cellular work to our previous animal work. And last but certainly not the least, when we co-treated the cells chronically for 24 hours with morphine as well as with BQ123, we saw a further significant decrease in these cyclic AMP levels. We were really not sure what we were going to see, and we weren't sure what to expect. But with both the doses of morphine that were used as challenge doses, the 10 micromolar dose as well as the 20 micromolar dose, both of these challenge doses were able to produce a significant decrease in the cyclic AMP levels as measured by the relative luminescence units using a plate reader when these cells were in the presence of BQ123 treatment. So again, these are, I think, really exciting findings, which help us to conclude that the cyclic AMP pathway is involved in the mechanistic effects of BQ123 on morphine. So we will be continuing these cellular studies, and we are actually in the process of establishing flow cytometry assays, and we are optimizing these to assess and determine the expression of both the mu opioid receptor as well as the ETA receptor, And the purpose of these current and ongoing studies is to look at the potential interaction between these two receptors in this particular cell line. Our future work will also include looking at beta-arrestin as well as GPCR kinases in these phenomena. And we thank you very much for letting us present our work today, and I'll be happy to answer any questions. Thank you very much.